Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Daniel bin Zamri. I have completed my internship program at Unit Bank Culture UITM Johor Negeri Sembilan, Kuala Pilah. First and foremost, let me start by telling you a little bit about the organisation. Unit Bank Culture, or UBK for short, was established in 2022 in UITM Kuala Pilah. It belongs to the microbiology department of the Faculty of Applied Science in UITM. The main purpose of UBK is to provide bacterial and fungal cultures for students, lecturers, and third parties outside of UITM. Next, we move on to the objectives of my task during my internship program. The main purpose of my task is to provide bacterial culture for biology and microbiology students for their laboratory practical experiments. In addition to that, I'm also responsible for bacterial culture preparation for the students' final year projects and the lecturers' research project, as well as third parties that require bacterial culture such as USIM, Matrix, or UPM. For the methodology of my task, I began with receiving a work order from the lab staffs that came in the form of hard copies. Next, I jot down the species of bacteria that is required, the choice of media, and any additional notes. After that, I gram stain the bacterial samples in UBK to identify the type of bacteria and make sure whether it is suitable for growth or not. Next, I prepare the required media such as nutrient broth, nutrient agar, or McConkey agar. After that, I begin the culturing process by streaking the bacteria onto the new plate using an inoculating loop. The streaking method may be either simple streaking in a zigzag motion to ensure uniform dispersion, or the 4-quadrant method to ensure single colonies. During the whole process, I make sure that I strictly adhere to the aseptic techniques. After that, I incubate the bacteria at 37 degrees Celsius for 24 hours. After the incubation period, I gram stain the bacteria once again to make sure that it is the right species. If I fail to identify and isolate the right species, I would have to start again from the beginning. Next, we will move on to results and discussion. Once the final product has been acquired and identified via gram staining method and observation of morphology under the microscope, the lab staff would pass it to the subsequent recipients. To mitigate the risk of contamination due to prolonged storage, the bacterial culture was subcultured at specific intervals, every 2 weeks for broth media and every month for agar media. This is crucial to maintain the viability and quality of the bacteria. In conclusion, the preparation of bacterial cultures is a fundamental process, crucial for various research and studies purposes. Therefore, preparing a quality bacterial culture requires strict adherence to aseptic techniques and precise methods. That's it from me. Thank you.